So far we've described the idea that individual molecules can self-assemble through non-covalent interactions to form a supramolecular polymer, which we might call a primary fibril. What happens if those supramolecular polymers can begin to stick together? Maybe through van der Waals interactions or solvophobic interactions. Well, if they do, they'll form bundles. And those bundles get to the stage where they can potentially entangle to form a sample spanning network, where there are points of adhesion between those different bundles because of solvophobicity and van der Waals interactions. And that sample spanning network, macroscopically, if you look at the material, means the material would appear to be a gel. And this is an image of an inverted sample vial, and you can see that this sample is sticking to the top of the sample vial when you invert it, and it has gel-like properties. And the idea is that you might take just 1% of your small molecule in 99% of solvent and be able to immobilize that solvent, forming a bulk gel. And as it says above, if supramolecular polymers are able to interact with one another, giving points of adhesion between them, then they are able to access a new class of material, supramolecular gels. And the really cool thing about these materials is that each step of the process is reversible, making them highly dynamic, highly tunable and programmable. A second example of a gelator is one that we work on a lot in my laboratory. This is a benzylidene derivative of sorbitol. So in the middle we have a sorbitol, which is a sugar, and we've made these two acetals where pairs of OHs have reacted with an aldehyde to install these acetal functional groups. And it's sometimes referred to as a butterfly surfactant because it has two wings, these aromatic rings, and the body made from the sugar. How does this assemble into organogels? You have hydrogen bonds between the sugar bodies, and you have pi-pi interactions between aromatic rings. And you can see here the butterfly stacking one on top of the other. So this is one molecule, two molecules, and you can see that will be 3 and 4 and so on, forming a stacked array of dibenzylidene sorbitols. You can see where the pi-pi interactions will sit, and you can see the hydrogen bonds between the bodies of these molecules. And this gelator is used for a number of things in the real world. Perhaps one of the big applications is if you buy a Right Guard deodorant gel stick. That is made of this DBS. And it's not really playing a very active role there, it's simply a gel. They put the active ingredients, the fragrances, the antiperspirants into that gel, and then you roll it under your arm and the gel transfers the active ingredients under your arm from the gel stick. And in this case, the solvents are alcohols. So again, it's an organogel that's being formed in this case. So to finish this little screencast on self-assembling gels, we're going to do a case study, and we're going to look at an example that's come from my laboratory. And this work was done by Ed Howe. You can see his picture here. I'm going to label him and put his name. And he did this work when he was an MChem student in my lab. He then went on to St Andrews University to do his PhD in supramolecular chemistry. And what Ed was interested in was using gels like the ones we make to do drug delivery. Okay? And in this case, he didn't just want the gel to be like the gel stick, where you sort of rub it on and the drug comes out. He wanted the gel to play an active role in controlling the delivery. So, this is the gelator that Ed decided to use. Now I want you to notice that this is just DBS. There's the sorbitol body. There's the aromatic rings. There is one difference. We have this acyl hydrazide functional group in place. And this makes the gelator more water compatible. This was an innovation developed in our lab in York, and so the gel that you see here on the left is not now an organogel or being formed in an alcoholic solvent, which is where traditionally DBS has been used by industry. This is now a hydrogel. It's forming the gel in water. And if you form gels in water, this is really important if you want to do things like drug delivery, because the body is an aqueous medium. It's also really useful for environmental applications in water, food applications as well, because you're using a non-toxic solvent. So this modification moves the gelator to work in a different solvent, okay? It also plays a second role. This is a base. And naproxen is a drug that we were interested in. I'll tell you why in a moment. And naproxen is an acid. And so what you do is you get an acid-base interaction. And how that looks specifically, in this case, there's your acid. And you're interacting with the base of naproxen through the lone pair on the nitrogen, interacting with that OH. And there's some proton transfer from acid to base. So this becomes partly O- and partly NH3+. So there's some electrostatics there as well. So we have the drug interacting with the gel. If we mix naproxen into the gel, it intimately mixes and it goes and sticks to the gel nanofibers. The drug sticks to gel nanofibers. And the really important thing is that the gel can control drug release. How does that happen? Well, when you're at pH below 5, you have CO2H, and the drug binds to the gel because the acid interacts with that amine. pH below 5 is what you get in the stomach. If you get pH 7 to 8, then the pKa means that you have CO2 minus. The drug is released by the gel. And that pH regime, particularly slightly basic, pH 8, is what you get in the intestine. And for those of you that know your human biology, when you take a tablet orally, it first of all goes into the stomach, then it goes into the intestine. And the important thing about drugs like naproxen is we want release in the intestine. The reason is naproxen is an anti-inflammatory painkiller. It's a painkiller. It's the same class of molecule as ibuprofen, and it causes side effects in the stomach. So if you can deliberately protect the drug in the stomach and release it in the intestine, you're protecting the stomach against some of the side effects of the drug. What we're trying to do is prevent side effects by using this smart gel formulation that traps the drug by binding to it at the pH regime of the stomach and will only release the drug where you want the drug to be released in the intestine, where it can be rapidly taken up into the bloodstream and be useful for the patient. So we're designing a molecule that self-assembles into a fibril, that assembles further in the right solvent because of the choice of this into a network. And because of the choice of this group, it has such sticky fibrils that it can bind to acidic drugs. We're not just looking at painkillers, we're looking at a range of different drugs, statins as well, using this kind of approach. And in that way, we're using the gel to achieve better drug delivery.